The moderator for this session two. Session 2 is addressing nutrient needs due to the loss of function in genetic diseases. And our moderator is Erin MacLeod from Children's National Health System. And Dr. MacLeod is a member of the planning committee. So I will turn it over to her for session 2.
Thank you, everyone. I think we had a great first session, so we're going to get things started by looking at specifically inborn errors of metabolism. So I think we have a good distribution of great experts here to speak on that. First, I'd like to introduce Denise Ney from the University of Wisconsin.
Good morning, and thank you for the invitation to present. PKU is characterized by a deficiency in the conversion of phenylalnine to tyrosine. It is managed with a low phenylalnine, or as we say, a low Phe diet. An individual with classical PKU often needs to restrict their Phe intake to 300 to 500 milligrams per day. That's about 10% of typical Phe intake. Nutrient requirements in PKU are unique due to its genetic basis that affects requirements for Phe and tyrosine. Medical foods are essential for nutritional management of PKU, but they affect protein requirements, nutrient bioavailability, and the gut microbiota. PKU is inherited as an autosomal recessive. It is caused by over 800 mutations in the phenylalnine hydroxylase gene, which is primarily expressed in liver. Tetrahydrobiopterin is an essential cofactor for phenylalnine hydroxylase. In 2009, the FDA approved a drug called Kuvan, which provides a synthetic form of the tetrahydrobiopterin cofactor. Up to 20 to 50% of individuals with PKU who have some residual phenylalnine hydroxylase activity respond to this cofactor supplementation with a decrease in plasma Phe, which allows them to liberalize or increase their dietary Phe intake. There's evidence for both causality and an intake-response relationship with respect to the toxicity of Phe in PKU. Phe intake that exceeds anabolic needs increases Phe concentrations in blood and brain and is neurotoxic, resulting in profound cognitive impairment in PKU that is not treated with a low Phe diet that starts shortly after birth. The diet was developed by Dr. Horace Bickle in 1953. And because he felt people would not believe the effect of Phe intake for someone with PKU, he made a movie of the first child treated with a low Phe diet. That's available on YouTube, and it's very compelling regarding the toxicity of Phe. Maternal PKU results in impaired fetal development and congenital anomalies if the mother does not strictly control her blood Phe levels during pregnancy. Current recommendations recommend lifelong treatment of PKU with the goal of maintaining blood Phe in the range of 120 to 360 micromolar. In 2012, the NIH held a PKU scientific review conference with data analysis from the Agency for Healthcare Research and Quality. They concluded there was moderate evidence for a threshold effect of a Phe level of less than 400 micromolar associated with IQs less than 85. So to bounce off of Dr. Brannon's excellent introduction, I believe the health outcome in PKU is the IQ within the normal range. The biomarker that's used clinically is blood Phe concentration. However, this is really just a surrogate for brain Phe concentration. The low Phe diet for PKU is adhered to fairly well in early childhood when rapid brain development occurs. However, lifelong compliance with the low Phe diet is very challenging. The diet has two components. All high-protein foods are eliminated. This means no animal foods, as well as high-protein vegetable foods such as seeds, nuts, and legumes. Then, for nutritional adequacy, medical foods supplemented with micronutrients are needed. There are currently two types of medical foods. Those based on mixtures of amino acids leaving out Phe. This was introduced in 1972 in the US, and a newer formulation using the peptide glycomacropeptide. This was introduced in 2010 based on research from my group at the University of Wisconsin-Madison. GMP, a 64 amino acid glycophosphopeptide, contains only a trace of Phe and is a prebiotic. It is found in milk within the K-casein micelle. During cheese making, the GMP is freed from the K-casein into the whey from which it is isolated. The Genetic Metabolic Dietitians International Organization, using the Delphi method, has developed dietary recommendations for PKU. They recommend 20 to 50% higher protein intake when amino acid medical foods provide the primary source of protein, as occurs in PKU. The evidence base for higher protein requirements with an amino acid diet reflect rapid absorption and oxidation of amino acids, resulting in reduced protein synthesis. Consistent with this, we have observed reduced postprandial blood urea nitrogen with GMP compared to amino acid medical foods. Improved growth in children with PKU fed protein at levels above the RDA has also been documented. This principle is relevant to GI conditions where amino acid formulations are used. For an individual with classical PKU, or a virtual absence of phenylalnine hydroxylase activity, their Phe tolerance, or their minimum Phe requirement, is the amount of Phe needed for protein synthesis to support growth and maintenance. From 0 to 3 months, Phe requirements for infants established in the first collaborative study funded by NIH. This study used an empirical approach based on food records and multiple Phe determinations for blood. These requirements parallel those of Foman and Filer to support growth of healthy infants. After 3 months of age, minimum Phe requirements are lower for PKU compared to the DRIs. Minimum Phe requirements of enterally fed infants determined with the indicator amino acid oxidation method are very similar to those noted in the collaborative study. The breakpoint of 58 milligrams of Phe per kilogram per day represents the point at which oxidation of the indicator amino acid, in this case lysine, reaches a plateau, indicating that the minimum Phe needed for protein synthesis has been met. Nutrient requirements for individuals with PKU above 4 years of age are poorly determined. There are several approaches to establish Phe tolerance, or the minimum Phe requirement for adults with classical PKU. Our approach was to use intensive dietary counseling, along with food records and blood Phe determinations over a period of several months. And when we did this, we noted that Phe tolerance increased from about 5 to about 8.5 milligrams per kilogram per day. This patient, an adult female with classical PKU, represents one of a case series of patients published by Dr. Erin MacLeod, our panel moderator. 8.5 is pretty close to Penchard's value of 9.1 milligrams Phe per kilo, determined in 10 adult males without classical PKU using the indicator amino acid oxidation method. Now, this level of 8.5 to 9 is about 1/3, as expected, of the most current amino acid requirements. That is the WHO 2007 report. They recommend 25 milligrams of a combination of Phe and tyrosine. Interestingly, in our outpatient clinical trial, we observed that adults with classical PKU consumed about 15 milligrams of Phe per kilogram. However, their plasma Phe levels were outside of the target range. I believe this is the real world of what an adult can really do following their low Phe diet in the community. We also observed that individuals with with variant PKU consumed about 22 milligrams of Phe per kilogram, and their Phe levels were within the target range, and that's pretty close to the WHO level of 25. Tyrosine requirements in PKU are poorly understood. Because tyrosine is an essential amino acid in PKU and also the primary substrate for the synthesis of the catecholamine neurotransmitters, it's a big concern. And we often see low tyrosine levels in PKU patients, and the response is to give them more of a tyrosine supplement, to add more to their diet. Our research has shown that prebiotic GMP improves the bioavailability of tyrosine in PKU. Despite almost 50% lower intake of tyrosine with GMP compared to amino acid medical foods, fasting plasma tyrosine levels are not different and within the normal range. We used metabolomics analysis to ask the question, where is the tyrosine in the amino acid medical food going? Plasma tyrosine levels are not different. And consistent with similar levels of dopamine, we see similar excretion of the primary downstream metabolite of dopamine, VMA, in 24-hour urine collections. The metabolomics analysis tells us that the tyrosine is being converted by the gut bacteria to potentially harmful compounds, in this case, tyramine and phenyl sulfate. The metabolomics analysis, and this was all done by Metabolon, can tell us this because it can distinguish between compounds that mammals can't make, but the gut bacteria can make. Tyramine is formed by decarboxylation of tyrosine. It's present in fermented foods. It's associated with headaches. Phenyl sulfate is a known renal toxin. There are a whole series of renal toxins which are synthesized by gut bacteria from tyrosine and tryptophan. And it's well known with the management of people with chronic renal disease to use pre- and probiotic supplements to decrease the gut synthesis of these renal toxins. These findings were emphasized by a study from Portugal which demonstrated increased mean blood tyrosine levels in PKU subjects using a couple servings of GMP medical foods per day. This was a 13-month study. PKU is likely associated with conditionally essential nutrients. For example, high blood Phe may alter metabolism, as has been observed with lower cholesterol biosynthesis. People with PKU have low cholesterol levels. Inflammation and oxidative stress are observed in PKU and likely affect nutrient requirements. And lastly, low levels of several nutrients are observed in PKU, especially children. Low bone mineral density and increased risk for fractures has emerged as a complication of treated PKU. Our studies demonstrate that both the PKU genotype and an amino acid diet contribute to low femoral bone size and decreased femoral strength using the PKU genetic mouse model. Studies in healthy subjects demonstrate that chronic ingestion of a high dietary acid load is bad for bone. This concept is especially important with age-related declines in kidney function. Bone provides a huge bicarbonate reservoir, and it's known that osteoclasts are activated by low pH. Bone assists the kidneys in the maintenance of systemic pH homeostasis. Amino acid medical foods provide a high dietary acid load. This is because they have a high content of sulfur amino acids, and in addition, many of the amino acids, for stability reasons, are added as chloride salts to these medical food formulations. We have confirmed this in a pilot study of 8 PKU subjects in a crossover design, where we determined that renal net acid excretion in 24-hour urine samples was 3-fold higher with ingestion of amino acid compared to GMP medical foods. In addition, calcium excretion is significantly increased outside of the normal range compared to GMP, with similar intake of calcium and bone-related nutrients. In conclusion, nutrient requirements for protein, Phe, and tyrosine are unique in PKU. This has the principles of nutritional management of PKU, I believe, have relevance to chronic disease. In particular, increased protein requirements with amino acid-based diets, as are often required in GI disorders. The gut microbiota, how do these unique nutritional management approaches affect the gut microbiota? In particular, nutrient bioavailability, the synthesis of both beneficial, such as short-chain fatty acids, but also potentially harmful metabolites. Conditionally essential nutrients with altered metabolism and inflammation or oxidative stress. Dietary acid load and bone health. Here's one that kind of goes back and forth. You have chronic disease that's crossing from inborn errors of metabolism. Very interesting. And lastly, even though PKU is a monogenetic disorder, one that we've had the opportunity to study pretty well because of funding from NIH, it still has tremendous genetic diversity that infers substantial variation in nutrient requirements within a population. I'd like to acknowledge my PKU research team and with colleagues both from Boston Children's Hospital as well as the University of Wisconsin-Madison. And, funding is really hard to come by for this inborn errors of metabolism area. I can testify to writing many, many grants. But I know we have representatives from the FDA here, and I would just like to acknowledge the FDA Orphan Products Grants Development Program. Orphan disease is less than 200,000 people in the United States, and this was instrumental in me being able to conduct one of the largest clinical trials done in the PKU population. I would also like to acknowledge some helpful input from Amy Cunningham of the GMDI. Amy chairs the committee that established nutrient requirements for the GMDI for PKU, and also Dr. Paul Penchards, who is the pioneer in developing the indicator amino acid oxidation method. Thank you for your attention.